Good afternoon to everyone, especially Dr. G. So for our video assignment, we will be talking about the type 2 secretion system of Vibrio cholera. So I'm Sweatling and
I'm Pravina. So without further ado, let's move on with the video. What are bacterial secretion systems? Bacterial secretion systems are essential for the growth of bacteria. Some secretion systems are found in almost all bacteria and secrete a wide variety of substrates like proteins and toxins, while others have been identified in only a small number of bacterial species or are dedicated to secreting only one or few proteins. Based on the diagram shown, there are 5 types of secretion systems in prokaryotes. As mentioned, we will be focusing on the type 2 secretion system, which is in pink, and it is under the sec-dependent category.
so type 2 secretion systems are widely distributed among gram-negative bacteria, but are especially prominent among the gamma proteobacteria. So many of the substrates secreted by type 2 secretion systems are degradative enzymes, such as proteases, phospholipases, and chitinases. So here we have an example of a proteobacteria, which is a gram-negative Vibrio cholera.
The biogenesis of the type 2 secretion system starts at the outer membrane complex, which is in light pink. Next, the inner membrane platform is assembled with components in light green, blue, and in light blue. Lastly, the pseudopilins are added to the outer membrane and inner membrane complex to form pseudopilins. Now, the system is complete. The 4 components of the type 2 secretion system can be distinguished with the pseudopilus, the outer membrane complex, the inner membrane platform, and the secretion ATPase.
The type 2 secretion system is responsible as a major virulence mechanism in bacterial infection to secrete extracellular proteins. So it is categorized under the sec-dependent pathway, and it is unique in its ability to promote secretion of large and multimeric proteins that are folded in the periplasm. So the pseudopilus is driven into the type 2 secretin complex by a screw-like mechanism to promote the secretion of substrate proteins like toxins and hydrolytic enzymes across the outer membrane.
Moving on to the type 2 secretion system specifically in Vibrio cholera, the type 2 secretion system mediates the secretion of cholera toxin, which is a major virulence factor of the Vibrio cholera. There are 3 distinct forms of the type 2 secretion system in Vibrio cholera based on the sequence characteristics of secretin. As seen in the diagram, the 3 forms are extracellular cap, the outer membrane domain, and the periplasmic vestibule.
So the mode of action of Vibrio cholera depends on its primary virulence factor, which is the cholera toxin, and it is responsible for all the symptoms of cholera, such as diarrhea, vomiting, and dehydration. So the bacterial cell will attach to the intestinal lining, it will colonize and secrete the toxins, which will cause an efflux of water and ions from the intestinal cell, and this will lead to the loss of nutrients and watery diarrhea. So cholera toxin is a multimeric protein complex and is nearly identical to the E. coli heat labile enterotoxin, LT.
The type 2 secretion system pilus is important for the mode of action of Vibrio cholera. It consists of long filaments that are bundled into highly organized structures from the expression of PUL genes. An alternative role of the pilus in secretion is to channel secreted proteins past surface structures such as capsules or past aggregated bacteria present in the biofilm. With regards to the previous slide on how it attaches to the intestinal lining, the Vibrio cholera moves to the surface and attaches itself to the GIT tract. This attachment is aided by the LPS and the extrapolymeric substances, EPS. The cells will colonize and secrete EPS, which helps in the biofilm formation.
So now we will be looking at the responses under various conditions. So several of the species that encode for the type 2 secretion pathway can colonize the human GI tract and also live in the aquatic environment, either as a free-swimming organism or as a member of biofilm attached to chitinous or abiotic surfaces which are all characteristic of waterborne diseases. So the expression of the type 2 secretion system is regulated by many environmental stimuli by a two-component regulatory system. So it involves the first component, which is a sensor kinase to detect and transduce the environmental signals into the cell, and a second component, which is a response regulator protein to control the expression of genes. So the exposure of Vibrio cholera to different environmental cues will either induce or block the transcriptional expression of type 2 secretion system. So we will be focusing on two methods, which is gene manipulation and quorum sensing.
The first method that we would be discussing is the gene manipulation. The genes for several of the substrates for the type 2 secretion system pathway are regulated by quorum sensing mechanisms, including exotoxin A and elastase. The removal of the entire EPS operon or the inactivation of the individual EPS genes or even the pill D will compromise the pathophysiology of Vibrio cholera. In addition to the loss of cargo protein secretion and virulence, the type 2 secretion mutants exhibit growth defects, perturbed cell envelope integrity, altered ion homeostasis, and induction of the extracytoplasmic RPOE stress response. The diagram shown shows the origin of replication, the selectable markers, and the restriction enzyme sites for Vibrio cholera chromosomes.
So as mentioned from the previous slides, RPOE is an endogenous factor which forms the promoter complex to positively regulate the expression of EPS. So the suppression of RPOE will lead to the inactivation of EPS transcription. So we can see in the diagram, when there is RPOE suppression, there will be no binding of RNA polymerase, hence there will be no transcription.
The second method that we would be discussing is the quorum sensing. For Vibrio cholera, the master regulator in quorum sensing is HepR. LuxO and HepR are the two main quorum sensing regulators for Vibrio cholera. Mutation of the LuxO results in the repression of the HepR, which constitutively blocks the TOXR regulon expression as well as inhibits the EPS transcription. This blocks the transcription of the toxin co-regulated pillars, TCP, and the cholera toxin, CT, along with motility, protease production, and the biofilm formation. By regulating the quorum sensing system, the expression of virulence genes can be blocked. The diagram shows the regulation of biofilm formation in Vibrio cholera by quorum sensing. This diagram shows the pathway where the repression of HepR can occur. This repression can lead to the blocking of the virulence gene's expression.
So the prevention and control of Vibrio cholera depends on the antimicrobial susceptibility. So first we will be looking at antibiotics. So antibiotics such as chloramphenicol, carbanicillin, and kanamycin can be used in the following concentrations and will lead to the repression of virulence gene expressions by inhibiting several biological processes of Vibrio cholera. So the table shows the different antibiotic sensitivity and resistance patterns in Vibrio cholera. And since Vibrio cholera can develop antibiotic resistance through prolonged exposure, synthetic biology can also be used as an alternative to overcome this issue, which will be continued in the next slide.
Next, we will be looking at a new age concept of using synthetic biology by using genes to program cells to become cellular antibiotic factories based on a CRISPR technology. This mechanism can be executed by the beneficial microbes that could sense and kill Vibrio cholera, where they could offer potential alternative to preventing and treating cholera. Synthetic biology techniques have enabled us to engineer the human microbiota for therapeutic purposes. For example, the lactic acid bacterium that is engineered to detect Vibrio cholera can be used for this mechanism. These sensor proteins have been further layered with a genetic inverter based on CRISPR technology.
So we've reached the end of this video presentation, and we can conclude that the type 2 secretion system plays a big role in maintaining the life cycle, pathogenicity, and the virulence of Vibrio cholera. So the severity of cholera infection depends on the genes such as enterotoxin, which interferes with host cell function, invasion proteins for the travel of bacterium through mucus of the GI tract, and pilus formation to allow for phage attachment.
We can also conclude that the virulence and the biofilm formation of Vibrio cholera can be regulated by gene manipulation and quorum sensing. Gene manipulation is carried out by suppressing the expression of EPS and the virulence factors, whereas quorum sensing is done through the regulation of LuxO and HBr to block the TOX-R regulon expression. Regardless of both mechanisms, they will both lead to the inhibition of EPS and the virulence gene. Lastly, the application of antibiotic and a new age concept of using synthetic biology can prevent and control the virulence of Vibrio cholera. In the end, we can say that we have gained a lot of knowledge through this assignment. We have learned to appreciate the systems that we cannot see. We have also been able to relate and apply the theories that we have learned from this module in this video assignment. Therefore, we would like to thank Dr. G for this experience. Thank you for watching.